DNA is a long strand of nucleotides that carries information. The information necessary to make all the proteins in the cell is carried in the DNA. Now, to copy DNA into RNA, which is an intermediary molecule, you take the DNA and you match up nucleotides, one for one for one. They match exactly, and it simply transfers the information from the DNA to the RNA. And the RNA is then translated into protein. And the way that happens is there's a special machine called the ribosome. It reads the RNA 3 bases at a time. 3 bases specify one amino acid. The next 3 bases specify a different one. The next 3 specify a third. And the amino acids get linked together, and you get a chain of amino acids. That makes a protein. But it's more complicated than that. The sequence specificity in the DNA gets turned into a sequence specificity in the protein, but then the protein has to fold into a 3-dimensional shape. And it's specific to that sequence, that protein. And it's the 3-dimensional shape of the protein which determines what the protein does. Now, we have 22,000 genes, most of which make proteins, but it's even more incredible than that. We used to think that one gene made one protein. Now we know that one gene, one stretch of DNA, can make multiple proteins. The variation can be extreme. There's one gene in a Drosophila that makes has the potential to make something like 38,000 different proteins. That's incredible. We don't get code from random processes. Just think of what would happen if you scrambled the information on a CD. Just let random signal be read into a CD. If you played it back, would you get music? In the case of DNA, the information there is digital and specified and has a function, and you don't get specified digital information from random processes.